So, we all know that DNA is super important. However, what do DNA molecules actually look like? How does their structure work? Well, first of all, DNA, or if we want to look at the longer form, or what DNA actually stands for. Then we're looking with deoxyribo. So DNA stands for deoxyribonucleic acid. So we've got D and A, and we now know that DNA is made up of these things called nucleotides. We know that this structure here that we've drawn with the phosphate, a sugar, deoxyribose sugar, and a base is what we call a nucleotide. Now, how does the structure of a DNA molecule actually look? Well, first of all, the first thing that we can look at is the primary structure of a DNA molecule. Now, the primary structure of a DNA molecule simply refers to the order of the bases. So we know that there are 4 different types of bases that can be present in a given nucleotide. The base can be adenine, thymine, cytosine, or guanine. And so that means we've got 4 different nucleotides that are possible. We've got a nucleotide that is possible with each of these bases. And so the order of these different nucleotides, the order that these different bases appear in a DNA molecule, is what we refer to as the primary structure. And is this primary structure that dictates the sort of proteins that are generated by our body. So that is the whole purpose of DNA, to control how our body produces various substances, in this case proteins. So that is the primary structure of a DNA molecule. Now, we can step a bit further back and have a bit more of a look at what we can call the secondary structure. Now, the secondary structure becomes a bit more complex. Now, if we step back and we look at our 4 different bases over here, we've got adenine, thymine, cytosine, and guanine. These, in fact, fall into 2 different categories of bases. So we can call this column on the left, we can call these types of bases pyrimidines, and we can call thymine and guanine. Sorry, we can call the thymine and guanine pyrimidines, while we refer to adenine and cytosine as purines. Now, these words sound a bit random, a bit odd. However, basically, what this means is that adenine and cytosine both happen to have an NH2 group on them. So they have this amino group on our purines. However, our pyrimidines both have a C double bond O group. Now, we know that here, if we would draw out our amino group in full, then we've got a very sort of unbalanced bond here. We know that nitrogen is a lot more electronegative than hydrogen, so we're going to have a bit of a negative charge here due to the electrons drifting towards the nitrogen, and a positive charge here. Now down here, oxygen is much more electronegative, so we're going to have a negative charge there and a positive charge here. So what this means is that the delta minus, or the small negative charge on the oxygen, is going to be attracted towards a small positive charge on these hydrogen atoms. And so that means that purines can undergo very good hydrogen bonding with pyrimidines. And so what happens is that if we have a base here, if we have one nucleotide here forming part of our DNA, part of one DNA molecule, then what we have is we're going to have another nucleotide here that's in the reverse direction. And so we've got another nucleotide here, and it just so happens that if we have an NH group like that, and a C double bond O group like that, coming from this base, and an amino group coming from this base, then we're going to get some very good hydrogen bonding across this gap here. And so if we have one base here, if we have one base forming part of a DNA molecule that's going up and down this side, and a base here that's forming part of a DNA molecule going up and down this side, then if we have one purine and one pyrimidine, then they're going to undergo some very strong hydrogen bonding. However, it's not just any purine and any pyrimidine. It just so happens that the base adenine bonds very strongly with thymine, and so adenine and thymine are always present opposite each other on two different DNA strands, and similarly cytosine and guanine are both always opposite each other on two DNA strands. And so our DNA molecules are in fact made of these two different strands, each with their bases paired up nicely so that we have adenine on one strand and thymine on the other strand. And as I went through in the video on nucleotides, the two strands have their ends in opposite directions. So we know that each DNA strand has two distinctly different ends. So if we're to draw in another nucleotide here, like so, and we have our deoxyribose here, and we have a phosphate here, and another base here, then we can see that this end of the molecule is the phosphate group end, and this end of the strand is the deoxyribose sugar end. Now, on the other side, the deoxyribose sugar end is actually going to be down the bottom, and the phosphate end is going to be up the top. So simply, if we have two different strands of DNA, and they're sort of undergoing hydrogen bonding with one another, then their ends are going to be at different orientations. So the phosphate group of one end is going to be with the deoxyribose sugar group of the other end, and that's going to allow this hydrogen bonding to occur much more effectively. Now, as a result of this hydrogen bonding between two adjacent strands of DNA, which are pointing in opposite directions, they have their unique ends in opposite directions, we get this sort of double helix shape like this. So if we have one strand of DNA given by the yellow line and one strand of DNA given by the light blue line, then we get these two strands of DNA wrapping around each other in what we call a double helix shape. So they're wrapping around here like that, round and around and around. And these hydrogen bonds between the adjacent bases are kind of like bridges across which bond the two strands together. So we can sort of view this double helix shape as a spiral staircase, where each of the DNA strands, each of the main strands, is in fact a handrail on our staircase, and each of these hydrogen bonds between the bases is a step. One step on our staircase, and that is the secondary structure of a DNA molecule. And so we get our entire molecule is made up of two strands of DNA which are pointing in opposite directions. They have their phosphate group pointing in opposite directions, and their sugar group, their deoxyribose group, up opposite ends, and they are bonded together in this double helix shape due to the hydrogen bonds occurring between adenine and thymine respectively, and cytosine and guanine. So that is the secondary structure. Now the tertiary structure of a DNA molecule is slightly different. So we know that in phosphate groups, our phosphate group has a structure that looks like this, and then we have two oxygens, both bonded to our deoxyribose sugar group on either side, and then we have a fourth oxygen coming from this middle phosphate that has a negative charge. That is what a phosphate group looks like when it's bonded to two deoxyribose sugars. So it's like we've got our phosphate group here, we've got our phosphate atom in the middle, we've got two deoxyribose sugars, one on either side, we've got a double bond oxygen here, and then the other side we have a negatively charged oxygen. And so that means that a DNA molecule as a whole has quite a significant negative charge due to all these phosphate groups which carry their own negative charge. Now that negative charge on our DNA molecule allows it to bond with this special type of protein called a histone. And a histone is a type of protein. And so because our DNA molecule as a whole has a negative charge, our chromosome has a negative charge, then it means that our DNA molecule can bond quite interestingly with this histone protein. And so what that means is that the DNA while we've got this DNA strand, which is in fact a double helix, if we draw our DNA as a strand like this, where this line is in fact a double helix shape, if we zoomed in on this line, we would see a double helix. However, if we just look at this strand of DNA, this double helix strand, then it's going to wrap really tightly around this histone. So we're going to get this really jam-packed. We get what we get as a result of the chromosome bonding with this histone is that we get a really tightly packed chromosome like this. And so that basically just saves lots and lots of room in our cells. We get our chromosomes packed into a very small space because their negative charge allows them to wrap tightly around this special protein called histone, and therefore saving lots of space and allowing them to pack much more efficiently in our cells. Now, when we reproduce, obviously, our DNA needs to be able to replicate itself so that we can produce new cells that have the same DNA. And so what happens when we're dealing with replication is that these two strands come apart, so these hydrogen bonds between the corresponding bases break, and then each strand, so what we get is we get rid of our double helix, and we now have two strands, each with the base coming off them, their bases coming off them, and these sort of act as a template for the new DNA, for the other strand, for two new strands to be produced. And the way that acts as a template is that we know that if we had, say we had adenine here, say this base here is adenine, then we know that this base here will have to have been thymine because there was a hydrogen bond between these two bases. Then as our body functions naturally to produce a new strand of DNA, our body can see that there's adenine here, and thus it will produce some thymine to bond with that adenine. And in the same way on this side, a new strand of DNA will be produced. A new strand of DNA will be produced here, and this, our body will see that, oh, here we've got a thymine base, and so our body will produce an adenine base. And in that way, all these bases match up. Our body produces two new strands of DNA that match up with the two existing strands, so that we have the right bases bonding together, and as a result, these two strands here will bond together in their own double helix, and these two strands here will bond together in their own double helix as well. And we'll get two DNA molecules that both look exactly the same to the original DNA molecule that consisted of these two strands here. So we'll end up with two DNA molecules, one there and one there, as these strands wrap around one another. And that is how our DNA is replicated. Now, if we want to have a look at a brief example of how these bases work and how this bonding works, then if we know that if we have the sequence on one strand of our DNA molecule looks like this. If we have our phosphate group, and then we have a sequence of bases that looks like. So we'll just use the first letter of our bases. Maybe our sequence of bases will be adenine, cytosine, guanine, thymine, and cytosine, and then at the other end is our deoxyribose sugar. Now the phosphate and the deoxyribose sugar aren't part of the primary structure, but we're simply using this to distinguish between the two different ends of our DNA strand. Now we want to work out what the sequence will be on the other strand of DNA, the strand of DNA that is bonded with this strand here. Well, we know that the two strands are going to have opposite orientations, so we know that this end is now going to be the deoxyribose end on our new strand, and this end will be the phosphate end. And we know that adenine always bonds with thymine, and cytosine always bonds with guanine. So simply, all we have to do is rewrite this. We're replacing A's with T's and C's with G's, and vice versa. And so what we end up with is this structure here. So simply we have to recognize that for every adenine, there's going to be a thymine on the other strand, and for every cytosine, there's going to be a guanine on the other strand, and the orientation of the strand is going to be reversed. So that is the structure of DNA. That's how it all works, and hopefully that has been helpful.